Welcome to this week's NeuroTools Webinar series. This week, I have a pleasure of introducing Dr. Amos Bairoch, and I'm sorry, I'm probably mispronouncing that.
No, it's correct.
Okay, he's a bioinformatician and of course a professor of bioinformatics at the Department of Human Protein Sciences of the University of Geneva, where he leads the CALIFO group at the Swiss Institutes of Bioinformatics, really combining bioinformatics, biocuration, and experimental efforts to fully characterize human proteins. And this is really the strength of Switzerland in bioinformatics. Until 2009, he actually headed up Swiss-Prot, which is one of the most well-known databases in the world, I think, and now has merged with UniPro. So in the 1980s, he also created ProCite and enzyme databases. These are, again, extraordinarily well-known data resources for the scientific community. He's responsible for the development of XPacy, really the world's first website dedicated to protein molecular biology. So currently, Amos's main activities are actually focused on the development of NextPro. This is a knowledge platform on human proteins. And of course, what we would love to know about as much as possible is Cellosaurus, because we use it in the RRID portal. So he's been awarded many distinctions, and they're very, this kind of just reads like a who's who, right? European Lastus Prize, the Otto Nageli Prize, the HUPO Distinguished Achievement Award, and of course he is an ISCB Fellow. We are very excited to hear about Cellosaurus. This is a knowledge resource, knowledge base of cell lines. So I would like to just hand this off to now to our speaker. Thank you all for joining us. And one other little point of note here, this is being recorded, so and it will be made available on YouTube after this is complete. Okay, thank you, and Amos, please take it away.
Thank you very much, Anita. Thank you for the nice introduction. So indeed, I will speak about the Cellosaurus, which in fact was not planned. I mean, it's a resource which happened to exist because, in fact, as Anita said, I was involved in development of knowledge base for proteins, Swiss-Prot and now NextProt. And we wanted to capture information on experiments and, of course, tag them to what experimental evidence there was for those experiments, and of course, which, if it was done in cell line, which cell line were used. And that's where I discovered that there was not really anything, encompassing all of the different cell lines which are used in labs. And this led to the creation of Cellosaurus. But I will go back to that in a few minutes. So let's first start about cell line in general. Now, cell lines, it's maybe obvious to say, but they're used by labs in academia and industry all over the world since the 1950s. And a number of them are acquired by a researcher from cell collection. And there are well-known cell collections such as ATCC in US, DSMZ in Germany, ECACC in England, RICON in Japan. But also, and that's also quite important, many other cell lines are transferred between labs. A lab will establish a cell line and then distribute it to collaborators. Now, of course, there is a number of varieties of cell line. Of course, cancer cell line are the most well-known ones, like with very well-known ELA cell lines, the first cancer cell line established in 1950s. But there is also transform cell line, embryonic stem cell, iPSC, hybridomas, and so on. So a lot of different type of cell lines. And then, as I said already as an introduction, I was looking for a resource on cell line, and there are resources which are based on catalogs from the collection which distributes cell line, but of course, those catalogs only cater for the cell line which are distributed by a specific cell collection. There are some list of cell lines on different site and lab page. There are Ontology, and I will go back to that, on cell lines. And there are many specialized databases that caters either exclusively or partially for cell line, but I will go back to that. So anyway, there is a world of information on cell line, and what was lacking was a single resource where you could basically start looking into it and then link to all of the different resources that exist on the cell line. Now, as I was telling you about cell line collection, and there is some, issues on the way the cell line collection present data. There is about 45 major entities in all over the world which are selling cell lines, and most of them are not savvy in terms of good bioinformatic practice. So, already 2 years ago, I drafted a number of minimal requirements that would seem to be very, very basic and obvious, but that at least each catalog should have an individual page for each cell line, because believe me or not, some, a catalog, just have a list of cell line on a single page and know where to get to a full page of information on a given cell lines. It would be nice that the URLs of those cell collection page be based on their catalog numbers, which often it's not the case. It would be also very, useful that people be aware of what are the new cell lines that those collections are distributing, but also, and that's quite important, the one which are discontinued, because it happens quite often that a collection or a company will stop selling a cell line for different reasons, but what happens generally is that this cell line just disappears from one day to the next from the website, and there is nothing you can do about it to get information about it, and it's like it fell into a black hole. And also, and we of course discuss STR profiles, but that those cell lines collection distribute the STR profile of the cell lines they're selling. Now, just visually, if you look at the current situation, on your right, you have this schematic of the Excel spreadsheet with those 5 requirements. And in green, you have things which are okay. In orange, it's more or less okay. Things which are partially okay. And in red, it's basically the things which are not okay. And what you can see is there is no cell collection which are compliant with those 5 rules. 6 are even 0 compliance to the 5 different rules. It's red all over the line. But to be more optimistic, since this situation can look like, things are getting better and better. And some cell line collection are trying to do, the right thing, creating, putting the information on their servers, making lists of discontinued cell line and so on. But there is still a lot to be done. And I think that the main issue there is they don't consider themselves as information provider, they sell product, cell line. And in fact, they never thought that they would have a role in terms of making sure that data is available and that people can go back later to what they were providing in terms of information. But things are getting a little bit better, but still the situation is far from being optimal. So that's for cell collection. Now, for ontologies, cell lines ontologies, there is quite a number of different efforts around cell lines and ontology. The biggest one is the CLO, the cell line ontology, which has 39,000 terms, which describe cell lines, including out of those 39,000 terms are 30,000 cell line terms, basically cell line names, which are then described with the 9,000 other terms in terms of what type of cell lines there are, which tissues they come from. So it's a well-organized resource. Problem is that it has a high degree of redundancy, as many entries for a given cell line as there are distinct providers. And so, and it's not complete at all, because if you take into account that it's redundant and that it has, a total of 30,000 cell line terms, there is already in Cellosaurus, and we go to it about 100,000 cell line, you see that a lot of things are missing. But it's very high quality in defining cell type and cell categories. And that's very, good in terms of as an ontology. There was an ontology developed in India by Molecular Connection, a company in India. It's no longer maintained, so I'm not going to really discuss this, and it was quite small. But then there are some other ontology which are not specific to cell line, but which cater to cell lines. The most well-known and biggest one is the Branda tissue ontology, BTO. And it has 2,000 cell lines. But of course, this is limited by the scope of Branda in that they, in fact, captures information which is useful for the annotation efforts in terms of enzyme. So they will only put in Branda the cell lines that they need, in BTO, sorry, the cell lines that they need for Branda. Then EBI has developed EFO, the experimental factor ontology. Again, this is not a cell line ontology. This is basically which cell lines are used in context of some EBI database and of ENCODE. So there is 1,300 terms which are, in EFO. And then, but that would be more, less, I would say more anecdotal. There is a VCGO beta cell, the cell genomic ontology, which has 50 cell line. And of course, the oldest ontology of all is MESH, and MESH has 24 cell line, which, they created, a number of years ago, and I guess they stopped after creating a few of them and didn't go back to create cell line terms. Now you see that this is not satisfactory, not in terms of the description of the cell type and cell category, because CLO does a very good job of that, but if people want to capture all of the cell line terms, it's not in the ontologies that they will find all of them. Now, before going to, inside the Cellosaurus, there are two things which I want to go through. One is naming issues, which is really a problem in the world of cell line. People tend to use short cell line names, and this is a disaster. I can give you here an example. You have 10 names of 2 to 3 characters, and those 10 names refers to 37 different cell lines. Now, that's obvious if you're going to use 2-letter codes or 3-letter codes, so there's going to be conflicts and collision. And what's worse is when longer names are proposed by people which establish cell lines, many others will abbreviate them and go back to having 2 or 3 letters code. So for example, you have this cell line called FG2C3A, which is okay, it's a derivative of FG2, but people just put in paper C3A, which is not okay. Now, unfortunately, there's only a few nomenclature rules which have been proposed. One for insect cell line, which is quite well followed, but that's a small niche in terms of the number of cell line which are catered by this group of people which work on insects, and one for embryonic stem cell and pluripotent stem cells also, which is not yet well enforced, but I think it's getting better and better. And now the journal Stem Cell Research, which publish 1-page or 2-page articles on iPSC, is using and asking authors to use this nomenclature. And of course, in addition to that, there is a huge number of spellings, misspellings in papers and also in catalogs of cell lines. Now, so next problem, which is worse, is a little bit exemplified by this cartoon, and that's a problem of cell line contamination. It's a huge problem. I'm not an expert into it. I will, in fact, in a few minutes, tell you who are the experts into determining if a cell line is contaminated or not. But basically, there's been a lot of articles in the last 3 or 4 years explaining what the problem is and the fact that it's costing a fortune in terms of wasted money because people have not used the cell lines that they should have been using, and that they report results and conclusion based on the wrong cell line. Now, people estimate extent of cancer cell line contamination from 20 to 36%, which is enormous. Now, the solution, in fact, and I will go through it, is in two steps. First, people should check if a cell line is known to be contaminated. And for that, there is, a group of people, which, a committee called ICLAC, International Cell Line Authentication Committee, which basically looks at every report, either published or unpublished, of possible contamination, tries to really go deep into the information to see if this contamination exists, if it basically touch a whole cell line or only some stock of cell line in different labs. And they publish a list of contaminated cell line on their website. All of those cell lines, as you will see later, are of course reported in the Cellosaurus, but ICLAC is really doing a tremendous work in terms of trying to follow all the leads, the different leads of people reporting problems with different cell lines. So I think that's really a top group, and which are really committed to try to help clean the world of contamination, of cell line contamination. But of course, having the list of cell lines which are contaminated is one thing, but what's the next step is to make sure that people, when they are using cell line, test to see if that's the right cell line or not. And that goes to the STR profile, which are short tandem repeats. So those are loci which are informative polymorphic DNA repeats in the human genome. You could have STR profile for other species, but most people are using them for human cell lines. Those STR, loci are the same which are used in forensic identification and paternity testing. And they can be used, and they should be used, to ensure the quality and integrity of human cell line. So there is an ANSI standard which make use of 18 markers, and there is basically different groups which are working to extend this, as I said, to other species. And people are working now for mouse and dog cell line to also have list of markers which will allow people to test also their mouse and dog cell line. Now, the good thing is that there are many companies and also academic labs which offer service to test for cell line identity. So you can send them your cell lines, they will do an STR profiling and report back if your cell line is what it's supposed to be. So they will authenticate the cell line, give you back the list of loci and the allele which are found. So a small problem with those companies which offer those service is that they keep their database of STR profile private. The best thing would be that everyone shares a STR profile, so that if someone happens to have a cell line which conflicts with another one, well, having as many STR profile as possible would be a good thing. And it's very difficult to get people to submit their STR profile, to publish them, and so on. So it's a very, big fight to get this information and put it in the public domain. But I will come back to that. Okay, so now, that, as I said, leads to the Cellosaurus, which is a knowledge resource on cell line. Its scope are all the different type of immortalized cell line, but also finite life cell line, when those like fibroblast cell line, when those are distributed and used widely, like the very famous MRC5 or WA38 cell lines, which are used for vaccine production. It deals with vertebrate cell line, and of course, with a big emphasis on human, mouse, and rat, which are the three most used species in the life science in terms of cell line, but also in vertebrate, insect, and tick cell line. It does not include primary cells, because primary cells which are established from a tissue are not cell line, and neither does it include currently plant cell line. So if we look at what you can find in Cellosaurus, of course, and that's where it started, and that's why it was called the Cellosaurus, for cell line thesaurus. I mean, it's to have at least the name and synonyms of all those cell lines. And this is also important, the common misspellings of those cell lines. But of course, since it's much more than naming as important information, so of course, one wants to know which category of cell line it is, the gender of the donor. One thing which will be added in the next release, which will appear in 2-3 weeks, is the age at sampling of donor. Currently, this information is not available, but it will be in 2 weeks' time. Of course, the species of origin, if it's from an animal, the breed or subspecies. Quite important, the site of origin for metastatic cancer cell line and sampling for non-solid tumor cell line. Population doubling time. And I will go faster because I'm not going to go through all of it. Microsatellite instability. So information about which disease a cell line originates from, either cancer or genetic disease. And that's also something new and which is being added is important sequence variation compared to wild-type genome of the species. So either somatic mutation or genomic mutation when it's a genetic disease. Of course, what gene have been transfected, if a cell line has been selected for resistance to a compound, which transformant is used to immortalize it, and so on. Now, last slide about this, also if it's registered in official list. And very important, what I told you before about cell line catalogs and cell line collection. If a cell line has been discontinued from a catalog, that's important. And going back to the contamination problem and misidentification, which are the cell lines which are known to be either totally or partially contaminated and or misidentified, and of course, as much STR marker as possible. That's quite important. Now, one important thing is that from the Cellosaurus, you can go back to a lot of different resources. So of course, you can go back to cell line collections and to a lot of those different database and resource which caters either for cell line or which include information on cell line, like for example, Cosmic for the somatic mutation cancer. I mean, you have here a full list of different type of resource, each of them with different specificities. Now, how does this data get into the Cellosaurus in a basically 3-tier system? A lot of the data is extracted from, cell line collection. Now, as I told you already, it's not easy to get information from cell line collection page. So I tried to do this in a semi-automatic fashion. So I tried to get data from the cell line collection because they know about the cell lines that they're distributing. A lot of manual curation efforts. So currently over 15,000 papers have been read, and the information on those cell lines have been entered in the Cellosaurus. And of course, what I would like to see growing, and I hope there'll be some people here listening that may do so, is to submit information, either published or unpublished, because a lot of journals don't let you publish a lot about the cell lines that you establish. And it would be nice that this data is not lost or kept into a lab book, but that it goes into the Cellosaurus. Here are some statistics. Currently, as today, it's a release that you can see on the web as 83,000 cell lines, but the next release will be over 100,000 cell lines. So as of today, that's the numbers from almost 600 species, but most of them from human, mouse, and rat, as I told you. About 60,000 synonyms, and as I said, 15,000 publications, which means about 83,000 references to those publications. And what's very important is to link back to a lot of resources, and so 200,000 cross-reference to 73 different resources. Also 30,000 web links, and I was telling you about STR profile. Currently, there is 5,500 cell lines with STR profiles. So there's still a long way to go before all cell lines have public data on STR profiles. Where can you get the Cellosaurus? Well, you can go on Expazi, which Anita mentioned at the beginning of the talk, and you can search inside the the Cellosaurus on the website, I mean on Expazi. You can download it and use it locally. You have 3 different formats available, either a structured flat file, or you can have it in OBO, but the OBO version, which is compatible with, of course, all type of ontology, does not have all of the data because OBO format is not meant to be really a knowledge-based format, so you won't find all of the data inside the OBO format, but you will find all of the data inside the XML format, and I would encourage people to use the XML version because, of course, that's structured, complete, and you have all the data in one file. And you can get it by FTP on the Expazi server, and from the, on Expazi, when you're in Cellosaurus, you have a link to the download page. Just some statistic of usage. I mean, it was put on Expazi in the mid-2015, so a little bit more than 2 years ago. Now there is about more than 1,300 sessions per day. I mean, in the last few weeks, it was more like 1,400. And soon there would have been 2 million page already page view. I mean, as you can see, 500,000 sessions and 300,000 users. Now, one important thing is where, I mean, so Cellosaurus is part of the Resource Identification Initiative, which I guess all of you which are listening to the seminar know very well because Anita is one of the founders and main, lead person on this, very important, effort. And of course, here I just put in one slide that RRI introduced the concept of resource identifiers, which are persistent and unique identifier for referencing literature research resource, like of course antibodies, organism, and tools. And I don't remember exactly when we started working together with Anita, but I think it's almost 2 years ago, also with cell lines, with the Cellosaurus. So as most of you well know, but probably maybe some do not, the identifier are prefixed with RRID for research identifier, and a second tag which tells what type of resource it is, like AB for antibodies, CVCL for Cellosaurus, and so on. And people can access and search those RRIDs at the portal, of SciCrunch. Now here, I just give an example of an ELife paper with RRIDs. And not only does this paper correctly mention the cell line, where they got them from, and the RRID, so everything is there, name of cell line, RRID, and so where they bought it. But it also says that they have authenticated their cell line by STR profiling and also looked at contamination by mycoplasma. So I would say that's a good example of a paper which allows people to know what has been done and can lead to a reproducible experimental system. So that's just one example. There's more and more papers which have RRIDs thanks to the effort of the community and specifically the resource, the RRII initiative. Now here, just as I give you some screenshots, 2-3 screenshots of the Cellosaurus on Expazi. Here it's a cell line which is contaminated, so it says in red, problematic cell line, contaminated. The parent cell line has been shown to be itself a derivative of another cell line. This one is a cell line which has been derived from a contaminated cell line. So as the cell line, parent cell line was contaminated, it inherits the fact that it is also contaminated. And of course, you see that you have the disease, which is associated with the cell line, that it's derived from a metastatic site, the species, the parent cell line, sex of cell, and so on, a reference. I can show you here another example with a very famous melanoma cell line, where you see that there is also some information on what type of omics work has been done on this cell line. So you see that the cell line has been analyzed by exome, proteome, RNA-seq, protein expression, SNP array, and so on. And there is more and more cell lines which have been studied by many different omics technology. You see a sequence variation here. It's homozygous for a BRAF mutation. And down in the at the bottom of the screen, you see links to all of the children's cell line of this cell line. So this cell line has been used to create other cell line with different resistance to different compounds or for different reasons. So you see this, this list of children's cell line. And last screenshot, because it's a bit boring to look at screenshots. You see here STR markers for a cell line. And what's important maybe to notice is that you have on the left column the name of the marker, and on the right one the values of the allele, but you can see that there can be conflicts because for two different reasons. One is that cell lines can be a little bit unstable, especially cancer cell line, and they will easily lose or acquire an allele. And you can see this with the third line, the third allele, D16S539, where some people have seen 11, 12, 13, some people have seen 12, 13, some people have seen 11, 12. Basically, probably this cell line adds those three alleles, 11, 12, 13, but it's losing one of them, or it's not. Some people which are redistributing are not, do not see the allele anymore. But it can be sometimes that there are conflicts due to measurement technologies, sequencing, or PCR methodology, and it's important to also capture this information. Okay, yes, it was a last screenshot, but there is still one which is more to show you cross-reference. So cross-reference to different places, cell line collection, resource ontology, and a lot of different type of resource. Now, what could or will be added to Cellosaurus? Two things in terms of tools or a distribution of the Cellosaurus. One is we want to create a tool to allow comparison of STR profile between cell lines. So you will be able to input your STR profile of your cell line and then search in all of the Cellosaurus to see which one are the most close in terms of the profile of their STR markers. Another thing is integration of some Cellosaurus data into TWiki Data. I will come back to it in about a minute. In terms of content, some people have asked that the Cellosaurus also deals with patient-derived xenografts, and probably it will do so, but not immediately, not in 2018, but probably in 19. And there is some information still to add, like the tissue of origin, using Uberon as ontology, and a lot of other information could be added, like integrated virus, translocation in cancer cell line, and biosafety level. Now, I was telling you about TWiki Data. I've started with a number of people which are quite active in Wikidata to try to model the information on cell line in Wikidata so that it will be possible to write a bot which will take Cellosaurus cell line and enter them into Wikidata. Of course, not all of the data will be in Wikidata. For example, STR profile are not really, something to store in Wikidata and other things. But the basic information on cell line, its name, its type, is it a cancer cell line, which disease is it suffering from, the species, and so on, could be modeled quite easily. And there is now, basically a framework to do so. And I hope to be able to have a student coming in April, which will work on this for a few months. And if she managed to do it, then we can probably enter all of the different cell lines which are in the Cellosaurus in Wikidata, which will be quite useful in terms of semantic integration with other life science resources. I'm getting to the end because I wanted to leave space for questions. Now, to thank a number of people, Elisabeth Gastiger, which is a developer at the Swiss Institute of Bioinformatics, and she, in fact, is a person which has worked on XPASI almost since the beginning of XPASI in 1993. So it's a very, very old website, and she is a person which has put the Cellosaurus on XPASI. Alain Gatteau is a developer in my group which has developed the tools which I use to generate the OBO and XML version of the Cellosaurus. And one person, the chairwoman of ICLAC, Amanda Cape Davis, because in fact I'm in contact with her almost daily to ask questions about cell line which I think are misidentified, and She does a tremendous work as a cell line detective, and that's in fact her handle also on Twitter, and as a Secretary General of ELife. And also, I want to thank about now 500 people which have answered my nagging emails when I asked them questions about some of the cell lines and to obtain additional information on those cell lines. And I will leave during the questions two things. You can, of course, send email at the email which is shown here to questions, updates, submission, or on the Cellosaurus, on the Expansive site on the top right of the screen, you have a contact link. And if you want to have news on Cellosaurus, see what's happening, but also on cell line in general, you can follow the Twitter handle Cellosaurus. And I will stop here and wait for questions.
Excellent. Thank you very much, Dr. Bairoch. That was a very informative webinar. I've also taken the liberty of typing in that email and your Twitter account into our group chat, and I'll also put them on the YouTube recording link as well.
Okay, yeah.
Great. So we do have two questions from Dr. Bandrowski, who has had to depart, unfortunately, but this will be recorded, so she will tap back into the YouTube video to watch these, I'm sure. And I think you touched upon this a little bit in one of your screenshots, but she was wondering if you could speak to the evidence that your efforts in Cellosaurus are improving rigor and transparency in the scientific literature, at least as far as reducing the use of contaminated cell lines. Would you mind elaborating on that just a bit?
Yes, well, I don't mind, sorry. No, I don't mind. Well, I'm not sure that the Cellosaurus is already having a big impact on that. I think, things are very slow, unfortunately. And the number of journals which are, asking people to put RRIDs is not yet, enormous. It's growing. So I think things are going in the right direction, but I'm not sure that, we could already see a statistical difference between the number of people using a contaminated cell line now and 2 years ago. I think far more than Cellosaurus, I think that there were so many articles and so many blog reviews or tweets on the fact that people need to be aware of cell line contaminations, that at least this is getting better, that people are aware of it. I know it because, just to give an example, as it was said to Anita at the beginning, we have a small lab working on trying to find the function of some human protein, and we use, of course, cell lines. And one of the postdocs in our group, in fact, was in fact not verifying, exactly which cell line she got from, the next lab down the corridor. And after having read, and it was before, I started Cellosaurus, after having read about all of those issues on cell line contamination, she started worrying and asked me if she could send her cell line to be tested, which I said yes, and of course. And I think that's, in fact, getting better because of those publicizing of those efforts, of those papers and studies showing the extent of the problem. It will get better, as I said, and I think that what's being done with the resource identification initiative is really, really important. But like everything important, it takes time to, get established. And but that's life. Things take a lot of time. We would all like it to be much faster.
I think that's an excellent segue into the second question we have. It's a little bit broader. How long do you think it will take to clean up the literature, and how do you think we best get there? It sounds like you're leaning towards believing the first step is the notification of the issue, which it sounds like you touched upon in your last answer. But do you think there's a timeline for cleaning up this literature?
Well, cleaning up new literature, you mean? Because,
sure, yeah.
It's not really cleaning the literature, it's cleaning the experimental problems, because in fact the literature only reports what has been done, and if what has been done is wrong because people are not using the right cell line, well, even if you know it from the literature, it still is that you cannot have a good conclusion from it. So I'm optimistic that people are going to be forced more and more by journals and by their grant agencies to make sure that they're using the right experimental setups, that they're really taking care of noting what they're doing and what they're using in terms of lab reagents, not only cell line, but of course antibodies. But giving you a timeline, incapable of giving you a timeline. I'm optimistic, but thinking that it will take time.
I will take the optimism, absolutely. And I know Dr. Bandrowski wanted me to put this comment out there. Discontinued cell lines, in a way, are similar to discontinued antibodies. With reference to antibodies, they're also removed totally by some companies with no landing page. The only place these things are able to be found is the antibody registry or Cellosaurus. So this really highlights why these resources need to exist for reproducibility in science. That is all I have in regards to comments and questions. Dr. Bairoch, thank you so much for joining us today. Is there anything else you'd like to add at this moment?
No, except thank you for the invitation, and I hope that some people will contact me with questions, update submission, because this is really precious to get feedback. And I hope so that there will be some new contacts thanks to this webinars. And thanks again for organizing it.
Absolutely. Thank you for joining us. And this will be on YouTube shortly. And so we do get quite a few comments on our YouTube videos, which I will forward to you when we receive them. At this moment, I am going to end the webinar. Thank you again for joining us, sir.
Bye.
Goodbye.